I'm McCavour. I'm the soil specialist at Perennia. And so what I want to talk about today is soil test interpretation, specifically for the grape industry. I'm really going to focus on looking at the soil test report after you've taken the sample, and what are the components of that. So basically, I would just want to go through every element of a Nova Scotia soil test report. And some of you may be familiar with it, some of you may not be, but talk about what each component is, why it's important, and what range you want to be in for grape production. And then talk about some management considerations as we go along. Before I talk about the interpretation, I just want to talk about some key points about soil sampling. So why are you taking a soil sample? It helps you make informed decisions on inputs required for economical and efficient crop production, helps you make informed decisions about any fertility or fertilizer or lime that you want to use, and it can also help you diagnose problem areas or problems within your block or field. So some key points. So one thing to think about for particularly for these perennial systems for vineyards is where you're sampling. So you want to make sure that when you're taking your soil sampling, you're capturing what your plants can potentially take up with nutrients. So you don't really want to be sampling in the center of your rows because you might not necessarily have your root system out there being able to take up those nutrients. So you want to make sure that you're getting a composite sample of whatever block or field that you're sampling at that time. You don't want to go out and take a sample in one single spot because that's not going to represent your entire field. And so one way to do that is just to do some zigzag patterns throughout your field. And sampling back and forth. You can skip a row, but the main thing is to keep your sampling techniques consistent when you go sampling. The depth is important as well. So typically you just want to go the 0 to 15 or 20 centimeters deep. Some people in these systems will go a bit deeper, but if you're going to do that, you can separate your samples and do a 0, 15 and a 15 to 30. However, that top 15 centimeters is really where you're going to see the bulk of your nutrients, as well as the recommendations from the soil test report are typically based on that 15 centimeter depth. So those recommendations may have to be adjusted if you're taking samples from a deeper depth. So you want to sample at least every 3 years. So it's really important to maintain and set up a long-term sampling plan and maintain records. So some of you may have older records, or if you're a newer producer, you may not have those old records of soil test report, but it's important to keep those so you can identify any trends in your soil. The other thing is to identify appropriate sampling zones. So just because you have a block doesn't mean you can't separate it further. So if you have a dramatic change in topography or soil type or something like that, or a change in crop variety, it might be good practice to separate those blocks further for your composite samples. The final point I want to make just about soil sampling is when to soil sample. So one of the main things to think about is that you're sampling at a consistent time every time you sample. And for grapes, a good time to sample would be that mid-August to mid-September range. And if you have any more questions specifically about soil sampling, we can talk about them at the end too. But what I really want to talk about today is the soil test interpretation. So after you collect your soil sample, you send it into the lab and get results back. And so the sample that you collect, the results that you get from the lab is only as good as the sample that you collect. So one important thing to mention is when you submit your sample to the lab, try and remember to put your crop on there, because if you don't, the recommendations at the bottom and these ratings here won't appear. So on the soil test, it has your pH, your soil organic matter, macronutrients in red or secondary nutrients, your micronutrients here in yellow, your cation exchange capacity, base saturation, and lime recommendations. So I'm going to go through each one of these quickly and discuss what it is and why they're important. And I'm going to spend a little bit more time likely on pH because that's one of the most important things in our acidic soils is to maintain a proper soil pH. And we'll talk about that a little bit more. So the first thing on your soil test report is pH. So it's a measure of soil acidity. it measures the active hydrogen ions in soil solution. So that's important. So those hydrogen ions that are actively available in that soil solution, and it's measured on a scale from 1 to 14, whereas acidic soils are considered below 7. So Nova Scotia soils are often naturally acidic, and there's natural soil processes that lead to that soil acidification, as well as there are anthropogenic factors that can further acidify soil. The other important thing I just want to mention is that pH is on an inverse logarithmic scale. So as your pH is going up, your acidity is going down. So for grapes in particular, so a lot of crops, there's a certain range you want to be within for optimal plant growth. There is a range, so if you're beyond that range on either end, that doesn't necessarily mean that your crop isn't going to grow. It just may not be doing as well as it could. And so here's a piece of a table looking at recommendations for grapes for Ontario as well as Northeast US. And what's important to show here is that it may change based on the variety. So it's really important as a producer to know what that ideal range is for your variety, because it may change as you go to different varieties within your production system. So really we want it above that 6 to kind of 6.5, even closer to 7 range, depending on the variety. So why is that so important? So many of you have probably seen this graphic here or something similar, which is showing the impact of pH on nutrient solubility. So you want those nutrients to be soluble so your plants can take them up. And oftentimes acidic soils can lead to nutrient deficiencies because things like phosphate, calcium, magnesium aren't as readily available in those acidic soils. So this graphic down here is an example of phosphorus. It is highly dependent on pH. And so you can see this range between 6 and 7. That's where you get the most relatively available phosphorus. And as you increase above 7, you get a lot more fixation with calcium, or or you go down below 6, you get more fixation with iron or aluminum. And so that's why it's so important to maintain that soil pH. Now, pH is not the sole factor that impacts nutrient solubility, but it is a large player. Otherwise, it's really important for microbial productivity. Similar to plants, microbes tend to have an ideal pH range where they're most productive. So this can impact nutrient cycling within your soil, particularly nitrogen is a microbe-dependent cycle. So you want to maintain a good pH to keep those microbes happy. As well as it impacts your fertilizer use efficiency. So similar to what I talked about with the solubility of nutrients, if you're applying fertilizer, for instance, phosphorus fertilizer, it's not 100% efficient. And as you increase in pH or get to those ideal pH ranges, that efficiency increases. So that's an important consideration, that if you're seeing phosphorus deficiencies at a low pH, it may be something to consider that you want to adjust your pH instead of just adding more fertilizer to compensate for that deficiency. So where is pH found on the soil test report? So it's the first thing. So there's kind of 3 different pieces of the soil test report that look at pH, and again, it affects many other things on that report. But we're looking at the pH, then there's this buffer pH, which is second, and then the lime recommendation, which is calculated by the lab based on these values. And so, as mentioned, the pH measures the active acidity. So basically what this means for producers is that it tells you whether or not you need lime. So in this soil test report, we're looking at low pHs. So this is one from a producer that was looking to produce grapes, but this was from before the vineyard would have been established. So this tells you that, yes, you need lime. You're below that ideal range for that crop. The buffer pH, it's an important value, but really it measures the reserve acidity. So the reserve acidity are the hydrogen ions that are potentially bound to soil. And as a producer, it's not vital for you to understand exactly what it means. What you need to know with the buffer pH is that the lab uses this value to calculate your lime recommendation. And so basically it estimates your buffer capacity, so the ability of your soil to change when lime is added. And so the last thing is the lime recommendation, and I just want to spend a little bit time on this because it's important to understand what that LRCaCO3 means here. So basically that is the amount of calcium carbonate required in tons per hectare to increase the pH to 6.5. So in this case, for this soil, you need 9 tons per hectare. This soil needs 7 tons per hectare. So when you're looking at liming, there's a few different considerations. So calcium carbonate, pure calcium carbonate, we consider to have a calcium carbonate equivalency of 100%. But different liming agents or different things that you might be applying to adjust your soil pH have a different capacity to neutralize the soil. So that first pH value is asking you if you need lime, but there are other things on your soil test report that you can look at to determine what type of lime you want to apply. And this is largely based on your accessibility to lime. So maybe the source for calcite, the calcium carbonate, as opposed to dolomite, which has magnesium in it, is more accessible to you. So that does play a role. But you can look at your soil test report and look at your magnesium concentrations and see if dolomite might be a better option. Dolomite actually has a slightly higher capacity to neutralize than calcite, so that's where you may need to adjust those lime recommendations. And so another example of that is some people use compost, seafood compost, things like that, or wood ash to adjust their pH. However, those different amendments have different capacity to neutralize pH. So if that's your sole goal, you may need to apply more or less depending on what that calcium carbonate equivalency is. And then keep in mind that these amendments typically have other things in them. So you don't want to be over applying something else for the sake of raising your pH. So through the soil test report, it tells you if you need lime and how much lime you need. But the other thing that is important to consider is lime is often slow acting, and especially in a perennial system such as a vineyard, you may not be able to incorporate that lime, where incorporation often leads to a bit faster acting because there's more direct contact with the soil. The other thing is that different soil types have a capacity to change, so that buffer capacity either quicker or slower. So a sandier soil may change quicker, may respond quicker to lime, but those effects may not last as long, whereas a heavier soil may not respond as quickly to lime, or you may not see that response as quickly to lime. So keep in mind that it can take up to 3 years for it to be fully effective. Not that if you apply lime 1 year, you may not see some benefits in the following year. It just, you likely, especially if your starting acidity is at 5, you're likely not going to see it go to 6.5 within a year. So keep that in mind. Finally, the last thing I'll say about liming and pH is that as you see in this soil test here, the lime recommendation is quite high. And I don't believe the lab goes above a 10 ton per hectare lime recommendation. But if you have a really high lime recommendation, you might want to consider split applying. So really my general rule of thumb is anything above 5 tons per hectare or 2 tons per acre. You don't really want to apply at one time. So you may have to spread that out over a few years. So I spent a lot of time on pH, but I think it's really important. And so the second thing that to look at on your soil test report is your soil organic matter. And your soil organic matter is really, of this report, your best indicator of overall soil health because it plays a role in many things in the soil. So it can help with nutrient cycling, it helps contribute to soil structure, increase water holding capacity, reduces the risk of erosion, increases biodiversity, and it also has these negatively charged binding sites that helps with nutrient retention, which we'll talk about a little bit more after. And so this kind of encompasses your physical, chemical, and biological properties of your soil, whereas the soil test often really kind of focuses on that chemical piece. So soil organic matter is really the combination of living and dead plant and animal matter in the soil. These are the major contributors. And so on a soil test report, it's that third line down and it's represented as a percentage. And so again, we have this little table here. So really you kind of want your soil organic matter to be somewhere within that kind of 2 to 5% range. And if it's below that, then you want to consider applying amendments or implementing practices that help build that soil organic matter. The next thing on the soil test report, or not on the soil test report, is nitrogen. So nitrogen is very important. It's often one of the limiting nutrients in Nova Scotia. And what I want to point out about the soil test report is that it's a measurement of nitrogen is actually not on there. You can request it separately, but oftentimes it's not very valuable to use for a recommendation because the nitrogen cycle is so variable and dependent on many things that if you take a sample and measure for nitrate, which is a plant available form of nitrogen, by the time you get that soil test report back and are able to make a decision on what you're going to do and buy fertilizer or whatever other amendment you're going to use, that nitrogen value may have changed. So what's important on the soil test report, though, is that if you put in your crop, it is going to give you a nitrogen recommendation, and that nitrogen recommendation is based on the crop. So if you can see here, these recommendations, the nitrogen recommendation doesn't change for these two samples, but the phosphorus and potassium recommendations do. So the thing that I want to talk about with the recommendations is that these are a guideline. And so it's a great place to start. However, you may need to adjust these recommendations based on if you have other practices that supply nitrogen to your soil, such as applying other organic amendments, or if you are incorporating a cover crop with legumes, you may need to adjust this nitrogen. As well as even just it may be, you may find that that application rate may be too high for what your specific requirements are for variety. So keep in mind that this is largely based on the crop. There's no varietal differences in these recommendations. So the next one is phosphorus. So I'll go through kind of the major macronutrients on the soil test report one by one, and then I'll talk about the micronutrients very briefly. But phosphorus is represented on the soil test report, and it does give you a recommendation down at the bottom, again, if you put your crop in. And so you can see that the recommendations for phosphorus change in these soil test reports. So this is still grapes, but it's in a vineyard that has been established. And so you can see that the values of phosphorus change here, and that's reflected in the recommendation. So phosphorus is largely not very mobile in soil, so the uptake of phosphorus is dependent on the root system. And it's often in its unavailable form. So we kind of looked at that chart earlier with the pH. And so the other thing that I just want to mention here when reading a soil test report is that it gives the value in kind of its fertilizer value. So if you want the value for just phosphorus, you actually have to multiply it by 2.29. And then if you're looking at recommendations from other places, so this is phosphorus recommendation for Ontario, northeastern US, it's in parts per million. So one thing I want to reiterate here is that if you get soil tests from different labs, they may have their analyses represented by different units, and they may be using different tests. So it's important to acknowledge that. And note that the recommendations may change slightly depending on the tests that they're using. So if you're comparing the tests, this is in parts per million, you want to convert that kilograms per hectare to parts per million, but also recognize that the actual test the labs is using, it might be different. So the recommendations may be slightly different. The next is potassium. Similar to phosphorus, it's represented in its fertilizer values. So if you want just straight potassium, you do have to convert it. Potassium is mobile in soil, so it can move with water. But one thing I want to reiterate here about soil sampling and the soil test is that you can take your sample and have the estimate for the relative abundance of potassium in your soil. However, if there are other management practices that are affecting this, such as if you go out there in the spring and your soils become really compacted, this can impact the uptake of potassium, but that might not necessarily be reflected in your soil test report. So that's also an important measure when you're looking at these recommendations. You may have to adjust it based on your management practices as well. And then we have calcium and magnesium. So they help with cell nutrition, stress response. They're important for photosynthesis. Typically with calcium and magnesium, we're getting a lot of that through liming. So if you have a good pH, a lot of times it's directly related to calcium, but not always. So you could still require calcium. And so there aren't direct recommendations for calcium and magnesium, but it does give you these ratings here. And so for the nutrient ratings, these are only available if you put in your crop. And so they give you a general idea of where along that low, medium to high or excessive spectrum that you see here, where your crop falls. And so these are tables that are available through the laboratory website, and it does give you a general range of where these numbers fall, but it doesn't give it specific to grapes. However, you can look at the tree fruit and get a general sense of where you fall for all of these categories. And these ratings are only given for those 4 nutrients. So next is your micronutrients. I won't spend a lot of time on this, but what I want to reiterate here is that a lot of the nutrients are given in parts per million, so be aware of the change in units, and also that your micronutrients often have a narrow range in which that you go from deficient to potentially toxic. So something like boron, so say you have this 0.3 to 0.7 parts per million range that you're looking for. The recommendation is a little bit different in Northeast US, but again, this may be as a result of different lab techniques. Then you can look if you fall within that range. Some of the detectable limits, so this is an example where the lab can't really detect anything, or at least the confidence of detection is below 0.5. Is enough. So anything below 0.5 just says below 0.5. So that's an important thing to note, that some of these, all of these nutrients do have detection limits, and some of them, and they differ based on the nutrient. So the last 2 things I'll talk about before we get into questions are the cation exchange capacity and base saturation. So really, when I'm looking from the lens of a producer, I'm looking at the pH, organic matter, your macronutrients, and making sure there's no anomalies in your micronutrients. And then I might glance at this cation exchange capacity. So it's defined as the total amount of exchangeable cations that a soil can absorb. So really, it's an indicator of the ability of the soil to retain nutrients. And so it's dependent on the number of negative exchange sites found. So you have your cations, such as calcium and magnesium, that are positively charged, that combine to these negatively charged exchange sites on clay particles and soil organic matter. So if you have a low cation exchange capacity, typically your ability to retain those nutrients is not as high. And this is common in a sandier soil with low organic matter. And so this cation exchange capacity can be difficult to alter. And really one of the only ways to do it is to increase your soil organic matter. So just to give you an example, these are some different soil texture ranges and their cation exchange capacity, but you can see that for organic matter, that cation exchange capacity is quite high. And this is just a visual representation. So these are just magnesium, calcium, potassium ions that might be bound to the soil. So in a high, say, clay-dominated soil with high organic matter, you're going to have a lot of these exchange sites. But in a sand-dominated soil with low organic matter, you're going to have fewer of those exchange sites. And this kind of leads into base saturation. So base saturation, I don't often use for recommendations, but it can have a place. And so base saturation is the ratio of these 5 nutrients here. So potassium, calcium, magnesium, sodium, and hydrogen on that cation exchange complex. So you can use it as an indicator of soil acidification. So if this value here, the hydrogen value, is much higher than all the other values, typically you have an acidic soil. But fertility recommendations should not be made solely on base saturation. The reason being is that there are some theories about ideal nutrient ratios. So for instance, calcium to magnesium might have an ideal ratio of, I think it's about 8 to 1 or 10 to 1. But you can have that ideal ratio while both values are still deficient. So it's important to make sure that you're reaching these thresholds that you want before you really dive into these nutrient ratios. And so because it does not represent the total amount of nutrients in your soil, only the amounts relative to these other nutrients. So just an example here. So in a, say, fairly neutral soil, maybe slightly acidic, you might have these hydrogen ions on this cation exchange complex. And as you get more acidic, you might have more hydrogen ions. So your base saturation, so the amount of bases, so calcium and magnesium compared to hydrogen ions, is going down. And then as you get more acidic, maybe some aluminum, which is also an acidic ion, can come on that cation exchange complex. So what's important to know for your cation exchange capacity measured in the lab is it may impact how you want to apply fertilizer. So if you have a really low cation exchange capacity, you have a lower capacity for those nutrients to be held in soil, you may want to consider applying less fertilizer at one time and really applying in those split applications. So split applications are often a good idea anyway, but especially if you have a low cation exchange capacity, low ability to retain those nutrients long term, it's definitely something you want to consider more. So just to wrap up here, I'm sure I'm at least close to half an hour, if not over. You want to make sure that you're taking your soil samples properly at least every 3 years so you can keep track of your soil sampling. And your soil test interpretation and your soil test report is only as good as that sample that you collect. So if you don't collect your sample properly or take care for sampling, it's going to be reflected in that soil analysis. So make sure you understand your soil pH and know how to amendment and take in those considerations. Increase your organic matter if necessary, but also critically evaluate your pH, your organic matter, those macro and micronutrients, and use that soil test as a guide. It is an awesome starting place. It's much needed, but you need to also make sure you're adapting for your specific system. And then finally, just wanted to recap on understanding cation exchange capacity and base saturation and where they play a role in soil test interpretation.
Caitlin's done a great job of introducing about soils and how important they are to follow and I'm just going to kind of build upon that, and hopefully my slides will make sense as we move along, talking about the tissue side of things. Over the years that I've been involved looking at grapevines, we have to kind of be like detectives. So we need to be always looking at what the patients in the field are like, the vines. And then we have to think about how the patient's lifestyle impacts on what we're seeing. So how much crop was present, the type of pruning we were done, the training system used, what was the actual environment, which is where soils come into play. And then we have to think about it kind of like a buffet system, in that we want to check the patient's diet. So we look at what's available to them. And that's where the soil sampling is absolutely critical. And as Caitlin has highlighted, The pH is probably your biggest starting point in terms of nutrient availability. But we do have the option that rather than just looking at the food or the source, which is the soil, we need to take a look at the patient itself. So we can do tissue sampling of grapevines. And this has been advocated by many over the years, and there's lots of information published about it. So why do people do it? Why would we take soil and tissue samples? The vines aren't growing well, or our yields aren't high enough, or we think we can improve the yields that we're getting. And the idea is to take these things and we want to be able to correct some issues before they become a serious problem. So if we see a trend that things are declining in our analyses, we can moderate or impact it by applying supplements before it gets into an absolutely critical level. But the one thing I will put across to people is that the laboratory samples are generalized, they're bulked, they're general information. If anything, I would encourage you that you want to take your own samples from your own site, and that many times you want to sample your best areas of the vineyard or your best vines for tissue so that you can get your own base numbers of what your targets should be. Labs work on a generalization level. Let's get a little more specific and start looking at your own vineyard and your own blocks and having your own data. Nutrients are highly influenced by a number of things. They can be present in the soil. The lab can give us a result and say it. However, the amount of water may limit the amount of nutrients that are available to the vine growth and the productivity we see, or how frequently it rained. If we go into severe droughts, many of those mineral elements are not available to the roots until there's adequate moisture level in the soil. And finally, if we look at it, the temperature has a huge influence in the way that the plant takes up. The higher the rate of transpiration or growth of the plant, the more nutrients it will pick up. The root hairs touch the soil particles, they can access the materials, and it will take it into the plant. And finally, if we look at the balance in terms of soil particles and uptake by the vine roots and really getting into soil structure, that can have a tremendous influence. And finally, as Caitlin has mentioned earlier, the cultivar varieties that we have, the types of clones, and the rootstocks that are used with those clones, especially on vinifera grapes, can have huge interactions. Some rootstocks are very efficient at nutrient uptake of things like potassium, and other ones are not. So we have to take that into account. The big elements, as Caitlin had mentioned before, nitrogen, phosphorus, potassium, magnesium, calcium, and sulfur. They're highly used by the plant. They're withdrawn in great numbers. The micronutrients, these are ones that are needed in small amounts. In some cases, you can run into toxicity if you jump too far. A classic example is boron. And the irony with it is that boron toxicity and deficiency look identical. So here is where taking laboratory analyses for tissue is really, really useful. Nitrogen, it's part of your canopy, it's the structure. If we think about when we add nitrogen to a yard and to our lawn, that's what greens it up and makes it grow quickly. In this case, on grapevines, when we run into nitrogen levels, that's where you see lots of canes, shoots developing, lots of wood development. We're in the business of growing grapes, not growing wood, so we have to really watch our nitrogen levels as we go along. And tissue sampling can test for nitrogen levels in the plant, and is not really as fluid as in soils. Soils, you don't get a nitrogen result, but you get a general recommendation. With tissue, you can be far more fine-tuned. The key to remember out of nitrogen deficiency, and people look at it often, they'll be very bleached vines, weak, very light tissue growth. Your shoot growth is shorter, your photosynthesis is negatively impacted, and you may run into early leaf drop in the fall. These are all signs of nitrogen deficiency. On the flip side, we don't want nitrogen excess, because if we see really dark, big green leaves, we've got way too much nitrogen present in our vineyard. And that's resulting in growing a lot more wood and staying vegetative than being fruitful. So keep that in the back of your mind. Phosphorus, as was mentioned, a person asked about excess phosphorus. In most cases, phosphorus highly influences root development of a grapevine. And it's really critical that it be there, but it's also associated with the flowering aspects. What's of note to some people is that it can influence wine quality, the levels of phosphorus in the plant, but it also has a huge influence on disease resistance within the plant. There are a number of biorational materials coming along to help ensure that your phosphorus levels and other things in the plant get most resistant to disease infection. If you're running into a lot of disease infection, you might be in a situation of deficient phosphorus uptake by the plant. If we have deficiency, our roots don't grow as well. photosynthesis declines. Magnesium leads to magnesium deficiency because phosphorus has been associated with magnesium transport within the plant, and you can get premature leaf senescence. The picture that you can see is actually an example of Merlot, where we went in, we did the tissue sampling, and we found out that the leaves were phosphorus deficient. And this led to other problems. Is one of the big ones. It's highly influential for sugar uptake and sugar translocation within the vine. It's kind of the key ones, but it's also associated with winter hardiness and the ability to deal with adverse moisture conditions. Potassium balance can manage excess water as well as deal with insufficient water, but it has to be there in adequate amounts. Where does it come into play? Again, stimulating root development, stimulating shoot growth. These are all things that can be key. If you're in deficiency, you're going to start to see the symptoms. There is a situation, if you're deficient in potassium, all of a sudden you don't seem to get the Brix levels that you were hoping for, or the overwintering of the plant gets influenced. Premature leaf senescence can come along, but you got to be aware, again, of the cultivar or clone, especially in vinifera, and the influence with rootstocks on how potassium is taken up. Magnesium is another one. It's part of the chlorophyll molecule. It's a very critical component, and it's highly influential relative to cell division and berry expansion and berry capacity to handle moisture. Deficiencies of it, again, like a lot of things, photosynthesis goes down, shoot growth goes down. You can result in some bunch stem necrosis that can occur and shatter, dropping of fruit. All can be related back to a magnesium deficiency level. And as Caitlin mentioned, in the soil, calcium, magnesium, potassium are highly interrelated on how they act within the soil taken up by the plant. Similarly, how the plant utilizes them are also influenced. Micronutrients have multiple roles. I put this slide in here just to show that people will talk to you about things about how critical a particular singular element might be. They're all pretty much interrelated, so that if you look at boron with shoot development and root growth, and do you get good fruit set? Yes, it's highly involved in those points, but the plant is cyclic in its nature in terms of accessing it and moving its plant. Zinc, it's talked about relative to bud hardiness and the overwintering, but generally more so where there's zinc deficiencies in place. Adding extra zinc has improved bud hardiness, but if your plant's well within the ranges of zinc content, throwing more zinc at it is not going to make it more winter hardy. Understanding how do things move in the soil. Nitrogen's pretty mobile in the soil, but phosphorus isn't. It can be occluded or bound on the soil particles, but once it gets into the plant, it can move fairly quickly. Similarly with calcium, it's not really mobile in the soil, but once it gets into the plant, same thing. It's slow to go. This is where sometimes foliar applications of calcium have helped, where you're noticing plant deficiencies in place. Relying on soil-applied calcium is not the only answer. You may need to start looking at some mobility. And this has been true not only for grapes, but for other tree fruit as well. Magnesium can move well in the plant once it's taken up, similarly with sulfur. And in some cases, sulfur is being used as a fungicide. So generally, sulfur has never been an issue with grapes, at least that I've run into. Boron is highly mobile in the soil. It's low to mediate within the plant available forms of it. Recognizing, yes, it can move very quickly in the soil. The key is even spreading a material over the soil profiles. Copper, iron, manganese, as you can see, are relatively low. Same thing with zinc in the soil. But again, the plant does not have a high demand for these particular things, and we can augment that with other practices rather than soil-applied materials. So how do you decide what you really need? Well, as we've started with, you start with the soil. You need to know what the food source is for the plant. And then you want to be able to go and look at, are there problem areas? Can you visually identify within the block where there's trouble? And you want to monitor those nutrient levels, and then you can adjust with fertilizer and lime. So the soil tells us what we have available for the plant to take up. The tissue tells us what the plant is taking up. And finally, by fertilization, we can modify the plant itself. Knowing what the plant is taking up, we need to make sure we find out what the issue is before we make a corrective action. And this is really important. Sometimes there's other things at play when you look at tissue sampling that can influence the results that you get. And I want to bring your attention to these. So what's a tissue analysis provide? Concentration in the tissue, they'll be variable with the tissue selected. and the time of season selected. So generally what we're looking at are mature leaves, but not old leaves or old rachis. And the nitrogen contact will float over the season. So you try and set yourself with a benchmark time for sampling. The dilemma is that the tissue analysis, they'll give you the results, but it's not taking into account what stress that plant has been under. If you're taking samples under extreme drought or extremely high crops, or whether you have recently pruned those vines or the leaves are in a shaded area, are all going to skew your tissue analysis and may not give you the actual answers you're looking for. And finally, what's in the plant does not necessarily tell you what's available to it in the soil. So we always look at these as companion materials or processes or analysis projects. Why do people apply fertilizer? Some people just throw fertilizer on because I've done it that way every year for so many years, and I just keep doing the same thing. Or if you like these little kids that have this little pop gun and there's a bear, sometimes wishful thinking is what you're going to do is going to fix a problem without really answering it. Or you've got a really good salesperson in the area that can talk you into things. I'll just leave that alone. Or finally, the lab recommendation may be made. And that one you need to take with a grain of salt. Again, laboratories are not looking at the actual plants and the vineyard itself. It's relying on analyzing what you send to them. So they develop their results on what you send. And if it's not a good sample, they're not going to give you good answers. Some people like to look at tissue sampling prior to bloom. Myself, I have not gone down that route. Looking at the people in the western parts of the United States, as well as other areas, nutrient levels prior to bloom are so highly variable and dependent on the weather that it's been there. Generally, though, what you need to look at is if you were going to go down this route, again, it's not one that I've done, but there are people who suggest it. You want to take the leaf opposite the primary fruit cluster, which is the very first cluster coming from the base of the shoot. At veraison, you need to switch your sampling. Some places have had where you would take the sample again at veraison in the same location. Those numbers get really skewed. What we've recommended is try to take it in veraison at the first mature leaf on the original shoot. You don't want to sample leaves that are on laterals. You want to keep it back to the primary shoot that was formed, be it a primary, secondary, or tertiary shoot, but on that original shoot, not the laterals that have broken off it. Recognize that tissue results are going to vary. Older vines will give you different responses. Similarly, different cultivars will give you different responses. So at the laboratory level, if you're going to take a tissue sample, all of the samples have to be from the same cultivar. So if I've got Merlot, Cab Franc, and then I've got Lacadie in a block, I would take a separate sample for Lacadie. I would take a separate sample for Merlot. I have to keep those split. I can't mix them because the results will be different. The time of the year of sampling, again, the numbers will look very different if you take them close to bloom versus veraison. I've tried traditionally to encourage people to take their samples at veraison or within the 2 to 3 weeks afterwards or the beginning of fruit ripening. The plant is at its most mature phase. It's used up all the vegetative activity that it wants, and it's in a reproductive fill phase. Make sure you've taken into account vine stress. You don't want to go in and sample if there's been excessive rainfall or if it's been under excessive heat. Or if you do, make sure that you keep that in the back of your mind when you see the results. Similarly, if the plants are under severe stress from diseases or insects, it can also modify the results you're going to see. And the last one is most people are applying pesticides, regardless of whether it's organic, biodynamic, or what we would consider conventional systems. Many times, copper and sulfur are components in many of the fungicides and can skew some of the results that you're going to see. This is an example of a laboratory sample that was done in the bloom, post-bloom phase. Nitrogen levels were quite high. If you go along and look, and then all of a sudden they're saying zinc is very low. Well, zinc is always very low in the bloom phase on most vines. They've given you a normal range if you look at the numbers and analyze them. They may be okay for your vines at your location. This normal range, again, was generalized out of a laboratory analysis. What I wanted to highlight here is that under that, the previous slide, so you see all this, and then I read the rider clause, as I call it, the out clauses underneath. It says these plants are low in magnesium, and it may be due to local issues in terms of soil, magnesium, soil potassium, pH, and drainage. Well, they haven't identified. Those are all saying these are different conditions that can really impact on your lab recommendation. So watch. Make sure you're not just treating and throwing without considering what is the original cause or likely cause of the reason for having such low numbers. Similarly for zinc, maybe due to low zinc availability, high pH, or excess soil phosphorus. Well, what happens if you have those? Then you need to look at modifying those to correct the situation, not the zinc issue. Similarly, high manganese, sprays, dust, again, lots of products. This one I found interesting that they recommend an application when they're deficient at this stage. For most people, by the time they get the results and apply it, they've had very little impact on the vines themselves. Fruit set, generally by the time they get this response back, they're already through bloom, and applying these materials have not dramatically changed what's going to happen to that plant. Similarly, they tell you to go to use it and then go sample 14 days later. I can almost guarantee that everything will be good to high because you've just applied them to the plant and it's in that tissue currently. But that also tells me that there's an issue. The plant hasn't taken enough materials up. So why? Get back to the root cause rather than treating just the symptom. So you get some numbers back from the lab, and looking at a tissue sample is slightly different than looking at the ones from the soil test. Remember, everything has to mesh together. All of the elements need to work in concert, kind of like a band. But if one's totally out of whack or out of tune in this band, then we need to figure out how do we get it back into balance. These are some numbers. I went into the 2022 Perennia Grape Production Guide, and I looked at their grapevine nutrients, and I've kind of come up with a revised chart for it. The chart in the book itself was based on West Australia. I think it's a little more important to take a look and try to modify these. So what I've done is try to blend the numbers from the Northeast United States, some materials from Nova Scotia as well as Ontario, and my experience in dealing with other people within the US and the researchers. I've been working on tissue and soil analysis for grapes for about 40 years now, and have found that these have been much better numbers to utilize and take a look with for tissue results. And the tissue sampling and the tissue analysis process, regardless of lab, is pretty similar. Soil tests are somewhat different, but the tests that they're using for the tissue analysis are fairly consistent. The petiole ranges, as I've had in here, I've updated again, trying to look at it. This is just a different way of looking at it. If you're looking in the bloom periods, if you notice an example at the top, nitrogen, very high nitrogen levels compared to what's at veraison. Fully expected. Bloom is a period of high active vegetative growth. I would expect that my petiole should be much higher. But if you notice many of the other materials, they're not radically different between bloom and veraison. The only ones that we've run into a little bit is starting to look at, okay, boron and a few things. Again, I warn on the boron, and I'll highlight that, is that boron is highly related to fruit set, but it can also inhibit fruit set if it gets too high too fast. We've had people, I've experienced with some boron recommendation, they followed it and continued to do it for about 4 years consecutively. And suddenly they were saying, well, I'm still seeing the same problems. When we did the sampling, we suddenly found out that they were in toxicity levels. So again, be careful. Too much of any of the micronutrients generally is not a good thing. So I have some numbers. I wanted to put this in perspective. An example there is when you harvest the crop, look at the tonnages that you're getting. So if you're taking it, if you look at it and you say, okay, I'm harvesting 4 tons per acre or per hectare. If I look at nitrogen, I'm only removing 10 kilos of actual nitrogen. The lab had recommended 40 kilos of actual nitrogen. Grapes generally want less nitrogen than what a laboratory will recommend to you. But again, they're generalizing on the soil and generalizing on the crop. I know that grapes, they've thrown in some base numbers, but generally they're looking at what I would consider excessive yield, which is actually leading to poor fruit quality. And the wineries and the processors are never thrilled at getting grapes that are totally out of balance when the pH and the titratable acidity is out of whack, often as a result of excessive growth situations. So I guess I've gone through the tissue fairly quickly, but it's It's the type of thing that you want people to look at these results that have a lot of experience in growing grapes and what targets you should be looking at. Most cases, a lot of recommendations come in that I would consider luxury recommendations. And in this era of trying to maximize the fertilizer dollar, you need to put on what you need, that's for sure, but sometimes you got to be careful that you're not adding too much. So as with a soil sample that I concur wholeheartedly with Caitlin, of soil tests, at least 1 out of 3 or 1 out of 4 years, definitely. I look at tissue sampling 2 out of 3 years. You don't need to do multiple samples in a single season. It's overkill, but routine annual ones, develop your own program. Make a good map for you and for others to be able to follow, to understand where the samples came from. Remember, the data over time is good for your site. No 2 spots in all of Nova Scotia will have identical results for tissue and identical plant response. So you need to start comparing that with your on-site observations of vine performance. Where do you fit on the spectrum of the analyses? Can you be at the low end of the range or the high end of the range? Where should you be is what you need to find. I always recommend at the very beginning, take samples from really good areas and then do your sampling from your poor areas. Now you've got numbers to compare. And again, they're from your site. That's why comparing and saying, okay, this is a poor area and doing against the rest of the province may not be your best benchmark. But if you can check it against some good spots in your vineyard, those vines are doing well for a reason. Get those numbers and start to target to that. Develop a plan, 5-year plan of sampling. Over time, you don't need to sample every block every year. We've often taken the farm and broken it into 3 segments and looked at it for soil 1 year in 1 segment and tissue in the other 2, and then just keep rotating over time. You will get a really good base level knowledge for your vineyard site. The other thing I will say is never guess. Make sure you really understand what the need is, and then work with your fertilizer from there rather than just throwing some out because you think it might be. Have it looked at. Nutrient applications, if you've looked at the price of nitrogen along other fertilizers in the last 5 years, they've skyrocketed. And the other part that I learned is that micronutrients in soil applications often add a huge amount of expense to the system relative to the total amount of fertilizer being applied. You may be able to do things just as well through foliar applications, or as an example with boron, including it in a weed spray strip and doing much better application, much more evenly over the block at a lesser cost and getting better bang for your buck. Foliar fertilizers are good when symptoms are visible, but they're a luxury when you don't notice any issues. And remember, if you buy, sometimes foliar products will say they contain, as I call it, everything: manganese, zinc, boron, everything under the sun. Why would you put it on if you don't need it? Try and get back to looking specifically what you need and why you need it. And the other part is walk your vineyard regularly. Use your own experience, use your knowledge, along with your senses, and you can tell a lot more than any paper results. And this is some of the places that I go to and what I've gone through many, many times to come up with the nutritional materials for tissue and plants, as well as experience over the time in developing a database here.